just letting the last people in. All right. Hi, hello everyone. Hope everyone is well. And today we're really excited to have Melisa here with us, and Nuria Melisa García. She's going to be talking about the feeding ecology of Mesozoic mammals, a biomechanical approach. Just before we start the talk, Melisa's talk, we're just going to go over the format for today's seminar. If you have been to previous talks, it's pretty much similar. But we have this 5 minutes welcome and announcements with a few slides. And then we move to Melissa's talk. That's going to last about 30 minutes, and which is going to be followed by a 10 minutes more or less moderated Q&A, so we can ask some questions. And after that, we're going to transition into an informal tea time, so we can talk about the talk and other stuff. And just a reminder that if you have any questions about the talk itself, you can send the questions via chat to questions PERCS host, that today is going to be Kate. And just before we advance to Melissa's talk, just a little bit of PERCS housekeeping. And at PERCS, we value the participation of everyone interested in the paleosciences. And because of that, please remember to abide by our code of conduct. If you haven't done so and you're seeing today's talk, please go to our website and do it. About muting yourself, please be muted during the entire talk. You shouldn't be able to unmute yourself during the talk, but if you do, please remain muted. Just a second reminder that if you have any question, you can send to Kate. She's a questions at PERCS host today. And if you also have any technical issues that you notice, please send the message to Kate as well, to the same questions at PERCS host. About captions, we do have some Zoom captions that you can choose to hide or show them by pressing the little CC button down there on your screen. And right now you're seeing PowerPoint captions, but during Melissa's talk, you should be able to see the Zoom captions. And I'd like to remind everyone that if you know any outstanding early career researcher in paleo, please go to our website and find the nomination nomination form that you can submit nominations to for us to invite people. And just another reminder that we take demographic information using a SurveyMonkey, and the SurveyMonkey link is going to be pasted in the chat right now. And we use demographic information even though you have filled this form before. It's useful for us, and it's anonymous and it's optional, and we just encourage people to do it when they see the talks. So finally, introducing Melisa. She did her bachelor's in biology at the Universidad Autónoma del Estado de Hidalgo in Mexico, and followed by a master's in paleobiology at the University of Bristol in the UK, and then a PhD in geology at the University of Bristol as well. And right now she's an honorary researcher at the same university. So thank you very much, Melisa. I think now we can take the screen share and start your talk.
Okay, cool. Thank you.
Have a great talk.
Thank you. Okay, so you can see that now.
Yes. Yes.
Perfect. Thank you so much for having me today. I am really excited to talk to you about what Mesozoic mammals were eating and how we can infer the behavior of these extinct organisms. And everything that I am presenting here today is the result of my PhD studies in Bristol. But before we dive into this question, what were Mesozoic mammals eating? I think that we should address a more basic question, which is, what are Mesozoic mammals? So the mammal part is easy. It is a concept that we are all familiar with. After all, everyone attending this seminar today is a mammal. In technical terms, mammals can be separated from other animals by a set of certain characteristics that we all share. For example, we all have hair or fur. We produce milk to feed our young. We have 3 middle ear bones, among other things. Mammals today are incredibly diverse. They range in size from the smallest of shrews to the biggest of whales. They are found inland, in the oceans, and in the skies. You can find them from pole to pole and in every ecosystem in between. Mammals today are so successful that the era in which we're living today, the Cenozoic, is often called the age of mammals. And with good reason. The Cenozoic has been a parade of weird and wonderful mammals, including giant ground sloths, dozens of elephant relatives, polar bear-sized hypercarnivores, rhino-like herbivores, among many, many others. The beautiful illustrations I have here represent a drop in the ocean of the astounding mammalian diversity witnessed throughout the Cenozoic. Despite its name, mammals did not actually originate during the Cenozoic. We can trace our origin to hundreds of millions of years ago, to an era called the Mesozoic, the age of reptiles. And of course, the most famous inhabitants of the Mesozoic were the dinosaurs. And to be honest, who doesn't want to study dinosaurs? I am sure that most of my generation of paleontologists are here today partly because we all saw Jurassic Park when we were kids. But even without Jurassic Park, you can tell that 19th and 20th century paleontologists were all more into dinosaurs than into Mesozoic mammals. Just take a look at this. The oldest mammal forms ever found are thought to be something like 200 to 230 million years old, roughly as old as the earliest known dinosaurs. And the first discovery of a Mesozoic mammal fossil dates perhaps as far back as the middle of the 18th century, a whole century before the first T-Rex was ever found. And yet, until maybe a handful of decades ago, Mesozoic mammals remained largely understudied compared to dinosaurs. And were always pictured as being rat-like, generalized nocturnal insectivores living under the shadow of dinosaurs. However, these past few decades, there has been a myriad of Mesozoic mammal fossil discoveries, including these exceptionally preserved specimens that have really helped turn around our understanding of these creatures. Now we know there were gliding mammals, fossorial mammals, arboreal mammals, even swimming mammals, a true diversity of locomotor modes. Mammals in the Mesozoic range in size from the teensy 5-gram microdocodon to the 14-kilogram Repenomamus giganticus. They lived in every single continent on the planet, and some of those mammals survived the end Cretaceous mass extinction and gave rise to the huge diversity of mammals we see during the Cenozoic. But let's get back to the main topic of this talk, the diet of these animals. So why is it important to know what mammals eat? Well, first of all, it allows us to know who is who in an ecosystem, and furthermore, it gives us clues about the environment itself. So, for example, let's say that a given place has 5 different mammals. Does this tell us anything? Well, no, not really. We can find 5 mammals anywhere on the planet. But what if I told you that this place has a number of pursuit predators and an even bigger number of long-legged herbivores built for running and with a preference for eating grass? In that case, we might think that these animals all live together in a fairly open environment, just like the Earth. But if I told you instead that a place has a high number of animals that love hanging out in trees eat plenty of fruit, and carnivores stalk and ambush their prey? Well, in that case, we might think we're dealing with a more closed environment, such as a rainforest. And of course, this is an oversimplification, but I just wanted to illustrate the point that it is important to know not only who we are studying, but also what they're eating and other things that go beyond the scope of this talk, like how they're moving and how they're interacting with others, if we hope to understand ancient ecosystems properly. So, how do we go about inferring diet in animals that have been extinct for millions of years? Well, we can do one of two things. One, we can look at their stomachs. However, the chances of stomach contents being preserved inside an animal for such a long time are tiny. In fact, we only know of one Mesozoic mammal in which this happened. Its name was Trypanomaimus robustus, and this beautifully preserved specimen is the only direct evidence we have of Mesozoic mammals eating juvenile dinosaurs. Here, it's pictured eating a young Psittacosaurus. So, we cannot really rely on stomach contents as they are so rare. The other thing we can do is look at their mouths. And this is going to sound painfully obvious, but we use our mouths for eating. So, surely the best clues for what an animal eats are inside its mouth. Particularly, teeth are one of the most important sources of dietary inferences in mammals. And we can clearly see why. Animals with different diets have differently shaped teeth. Teeth are one of the most commonly preserved body parts in the fossil record, and as such, are very commonly used to infer diet in fossil mammals. Mesozoic mammals are no exception to this. But teeth are not the only mouth parts that can evidence what an animal eats. The lower jaw plays a hugely important role in feeding, and for a long time, jaw morphology, that is the shape and structure of the jaw, has been linked to diet in mammals. For example, carnivores such as cats have short jaws for a more forceful bite. Their blade-like teeth are in line with the jaw joint, which allows them to have a scissor-like bite. And they have a large attachment area for the muscles that will help them pin down their prey. On the other hand, large herbivores, such as horses, have long jaws that allow them to reach down to the ground and cut grass. Deep jaws to house their very tall teeth used to resist abrasive grasses. And a large attachment area for the muscles that will help them process plant matter. And this is where my main research interest lies. What can the jaws of Mesozoic mammals tell us about their diets? In particular, their jaw shape and function. So let's start with jaw shape first. Jaws are one of the most commonly preserved elements of Mesozoic mammals, which makes them a great study object. And as you can see, they come in all shapes and sizes. However, on their own, they're not really telling us much. We need to compare them with the jaws of modern mammals of known diets if we want to make sound inferences. And this is true for all of the analysis that I'm going to talk about for the rest of the seminar. So I am not the first person to use jaw shape to infer diet in Mesozoic mammals. A few years ago, Rosnicol and Polly analyzed the jaw shape of a number of modern small mammals and found that overall, modern insectivores and carnivores have different jaw shapes to modern herbivores. However, they found no difference between insectivores and carnivores. Afterwards, they included Mesozoic mammals into the mix and found that non-multituberculates are similar to modern insectivores and carnivores, whereas multituberculates were more similar to modern herbivores. And I know you might be asking yourselves, what is a multituberculate? Well, multituberculates were a hugely successful order of mammals that first originated during the Jurassic and survived the end Cretaceous mass extinction. They are rodent-like in appearance, have complex teeth and jaws, and unique chewing movements. Because they are so different from other Mesozoic mammals, we decided to exclude them from our study and focus only on mammals with relatively flat jaws. I scoured museum online databases, scientific papers and books, and even got the chance to visit a number of museums and collected photographs of the jaws of 45 Mesozoic mammals and 17 modern mammals. Most of our modern mammals are under 5 kilograms because most Mesozoic mammals were small, but we also included a few larger ones up to 14 kilos. Then I captured their jaw shape using a technique called 2D geometric morphometrics. For this, I positioned 7 fixed landmarks in all of my specimens in easily identifiable points across all specimens. For example, the tip of the jaw or the top of the coronoid process. Afterwards, I positioned 58 sliding semi-landmarks that would help me capture. Once I had jaw shape data for all of my specimens, I needed a way to compare them to each other. So I used the principal components analysis to explore jaw shape differences across my specimens. And here is what I found. So we can see here a plot of all the different specimens of modern mammals that I analyzed. However, this is not really telling us much. We have to know who is who in this array of blue dots. So that is what I did. And I noticed that on the left-hand side of my plot, I had animals with short jaws, and on the right, animals with long jaws. On the top of the plot, you could see animals with tall ascending rami, which is the structure indicated here in red. And in the bottom, we could see animals with short ascending rami. If we then look at what all of these animals are eating, we'll realize that herbivores have short jaws and tend to have tall ascending rami. Insectivores have longer jaws, and the height of their ascending rami varies quite a bit. Carnivores have short to medium jaws and a diversity of ascending rami heights. And finally, omnivores plot all over the place. If we then add Mesozoic mammals into the mix, we'll start to see some very exciting results. Mesozoic mammals that are thought to have been insectivores based on tooth type, body size, tooth wear data, etc., largely plot on the morphospace of modern insectivores. And the same is true for most carnivores. We have a couple of putative omnivores that plot in very different parts of the graph, one near herbivores and the other one near insectivores. And finally, we have a large number of Mesozoic mammals that no one has offered a possible diet for. Most of them plot near modern insectivores, but maybe a couple plot closer to carnivores. So, principal components analysis is a great tool, but it is largely exploratory. So we wanted to use a classification tool. And for that, we used a phylogenetic discriminant analysis. What is a phylogenetic discriminant analysis, you might ask? Well, here is how it works. You start by telling the software, this is what a modern herbivore jaw looks like. And this is what a carnivore jaw looks like. And finally, this is what an insectivore jaw looks like. Also, here is how all of the animals in my sample are related to each other. Now, here is a jaw of a Mesozoic mammal. Based on everything that I just told you, can you tell me if this is a herbivore, a carnivore, or an insectivore? So we did that for all of our Mesozoic mammals. And the analysis classified Mesozoic mammals with about 89% confidence. So bear in mind that there is room for error here. Most Mesozoic mammals in our analysis were classified as insectivores, including most early mammals such as Morganucodon, and like the tiny Microdocodon, as well as a number of other groups, including the monotremes and eutherians in our sample. Monotremes are the group the modern platypus and the echidna belong to, and eutherians are the groups humans, dogs, cats, elephants, etc. belong to. So Mesozoic mammals were classified as carnivores, including the Eutheroconodontids and Gobiconodontids. This is the group the dinosaur-eating mammal Rapinomamus belongs to. Most Metatherians were also classified as carnivores. Metatheria is the group that modern marsupials, such as kangaroos, possums, koalas, etc., belong to. Finally, a couple of species were classified as herbivores. One of them, Haldanodon, was not very confidently classified as a herbivore, and the other one, Bin celestis, is a very particular animal, which I'll come back to in a sec. But let's move on now to what jaw function can tell us about the diet of Mesozoic mammals. And for this, we're going to treat the mammalian jaw as a lever. So what is a lever? It is a beam-like object rotating around a fixed point called a fulcrum. The purpose of a lever is to transmit energy from one end of the beam to another. Levers transmit a force input into the system to provide a greater output force to displace a load. This is known as leverage. And there are three types of lever, defined by the position of the fall, effort, and load relative to each other. Here are some common examples. A pair of scissors is a first-class lever. A wheelbarrow is a second-class lever. And a hammer is a third-class lever. In particular, I want to focus on third-class levers, in which the fulcrum is on one end of the beam, the load is on the other end, and the effort is in the middle. And I want to focus on this type because the mammalian jaw often works as a third-class lever, in which the jaw articulation with the skull or condyle acts as the fulcrum. The jaw closing muscles, also known as adductor muscles, generate an effort to pull the jaw upwards, and a load is located towards the anterior end of the jaw. A way of describing the leverage of a system is by calculating its mechanical advantage. In this case, we did so by dividing the length of the in lever by the end, which is the distance between the fulcrum and the effort, by the length of the out lever, which is the distance between the fulcrum and the load. The mechanical advantage of the jaw represents a compromise between a forceful bite and a quick bite. On the left, you can see a jaw with high mechanical advantage of the temporalis muscle, and this animal has a powerful but slow bite. On the right, you will see a jaw with low mechanical advantage. This animal has a weaker but quicker bite. So what we did was measure the mechanical advantage of the main two jaw closing muscles, the temporalis and the masseter. And here you can see a diagram of how we took those measurements. In pink, you can see the in lever measurements of the temporalis above and the masseter below. And in blue, the out lever measured at the tip of the jaw. So, what did we find? In general, modern mammals tend to have, modern insectivores tend to have low mechanical advantage values of both the masseter and the temporalis. Because insectivore jaws are so long, their out levers are comparatively much longer than their muscle in levers, resulting in these low mechanical advantage values. But this works well for insectivores because they rely on speedy bites to catch insects, and also because they need long snouts. They can get into holes to look for insects. Carnivores also have low mechanical advantage values of the masseter, but tend to have higher temporalis mechanical advantage values. The temporalis mechanical advantage values tend to vary quite a bit because among carnivores, those who eat almost purely meat, like cats, tend to have shorter jaws than those who have a bit more varied diets. Finally, herbivores tend to have higher mechanical advantage values of both the masseter and the temporalis. And this is very interesting because, as I said before, it is commonly thought that carnivores are the ones with the big temporalis muscle and that herbivores are the ones with the big masseters. The carnivores in our sample tend to follow these rule very well, but not the herbivores. Remember, they have high mechanical advantage values of both the masseter and the temporalis. So what is going on here? Well, this idea of herbivores with huge masseters and tiny temporalis muscles comes from big ungulates like horses. But it seems like in small mammals, both carnivores and herbivores have big temporalis muscles that help them resist dislocating forces when they're biting at the front of the jaw, either when dealing with struggling prey or biting hard plant material. But what about Mesozoic mammals? Well, in general, Mesozoic mammals plod where it's expected of them. Putative insectivores have low mechanical advantage values of both muscles. Carnivores have relatively low masseter mechanical advantage values, but higher temporalis mechanical advantage. The omnivores in our sample seem to have similar values to modern herbivores. And most Mesozoic mammals with unknown diets are similar to modern insectivores. You might have noticed that some Mesozoic mammals have higher than expected mechanical advantage values of the masseter. And all of these are early mammals. So why might this have happened? Well, the earliest mammals had very anteriorly positioned angular processes, which means that when I was taking my measurements, the in lever of the masseter was very far forward, thus resulting in higher than expected mechanical advantage values. Early mammal jaws look like this because they still retain postentary bones at the back of the jaw, which in later mammals separate completely from the jaw bone and become the middle ear bones we have today. So, in general, I've shown two things. One, that jaw shape and mechanical advantage are good proxies for diet in Mesozoic mammals. And two, we've corroborated that most Mesozoic mammals were probably insectivores, but some were carnivores. But before I move on to the next thing, I want to highlight some mammals I found interesting. First of all, the swimming mammal Castor rocata is thought to have eaten fish, but here it plots right in the middle of modern insectivores, so maybe of soft aquatic invertebrates. The early mammal, Agilodocodon, was originally proposed to have been a herbivore eating tree sap, but a later study found no evidence for this and instead suggested that it was an insectivore. Our study supports the hypothesis that Agilodocodon was an insectivore. Finally, the South American mammal Vincelestes is a very interesting case. Previous studies have suggested that its tooth wear is reminiscent of modern carnivores, but its jaw shape is very herbivore-like, according to our study. So maybe Vincelestes was an omnivore. So up to now, we have been looking at jaws, both in shape and function in 2 dimensions. But of course, jaws are 3-dimensional, and thus I want to analyze them as such. To do that, I used an engineering technique called finite element analysis, or FEA for short. FEA has been used for a long time in engineering and in orthopedic science, but it's been adopted more recently into paleontology. In this case, we're going to use it to analyze the performance of the jaw at the moment of biting. And there are 2 great things about FEA. First, it allows us to break down a really complicated shape like a jaw into smaller, simpler shapes that we can easily analyze. And second, it allows us to simulate the way the animal was biting. It has information on the material the jaw is made of, the direction and force the muscles are pulling with to close the jaw, and the areas where movement is constrained. And in the end, it tells us how much stress the jaw was submitted to at the moment of biting. And by analyzing that, you can tell how feasible it was for an animal to eat different things. Under identical loading conditions, you would expect an animal that eats soft food like insects to have a relatively weaker jaw than an animal that eats tougher food like meat. Therefore, a relatively weaker jaw would exhibit larger areas of higher stress than an animal with a relatively stronger jaw. So I used a subset of my previous dataset to analyze jaw function using FEA. For this, I used 23 Mesozoic mammals and 30 modern mammals. All the models that I built were the same size and were loaded identically, so that any resulting differences were due solely to the differently shaped jaws. All models were constrained at the jaw joint and at the first lower molar, and both the masseter and the temporalis were modeled. In these heat maps, cooler colors like blue represent areas of lower stress, and warmer colors like red represent areas of higher stress. And here I do want to apologize to any people in the audience with color vision deficiencies, as finite element heat maps are not very accessible, but I really hope that we can change that soon. For now, allow me to describe my results. So I found here that insectivores had large areas of medium stress and some substantial areas of very high stress near the jaw joint and along the ascending ramus. When we look at carnivores, we can see some differences between hypercarnivores, that is, carnivores whose diet is almost composed of meat, composed of meat, and mesocarnivores, which also eat insects, fruits, and other things. Hypercarnivores tend to have large areas of very low stress, whereas mesocarnivore jaws tend to be a little bit more stressed. Herbivores tend to have large areas of low to very low stress, and some areas of medium stress. Finally, there's a diversity of stress patterns among omnivores. Insect-dominated omnivores have very large areas of medium stress and big patches of very high stress, whereas plant-dominated omnivores and omnivores with an important component of meat in their diets have very large areas of very low stress. The vast majority of Mesozoic mammals have large areas of medium stress and patches of very high stress, very much like modern insectivores. But a few Mesozoic mammals have large areas of low to very low stress, very much like modern hypercarnivores. If we perform a principal components analysis of the stress distribution of these jaws, we'll find that at the top, we have jaws with larger areas of high stress, and towards the bottom, jaws with larger areas of low stress. If we see where the different dietary categories are plotting, we'll find insectivores towards the top, herbivores towards the bottom, and carnivores somewhere in between. Some of the carnivores that plot towards the bottom right are hypercarnivores, and those closer to insectivores are mesocarnivores and piscivores. Once more, omnivores plot all over the place. However, in this case, the omnivores located towards the top have insect-dominated diets, whereas the bottom ones incorporate a lot of plant matter and meat into their diets. And once more, what about Mesozoic mammals? Well, putatively insectivorous Mesozoic mammals plot towards the top most near modern insectivores. Most carnivores plot towards the bottom of the plot as they have large areas of low stress, similar to hypercarnivores and some herbivores. The only putatively omnivorous Mesozoic mammal in our sample plots near the insectivores. And most of the mammals with unknown diets plot near the insectivores as well. So let's put everything together and answer the question, what can jaw's shape and function tell us about the diet of mammals? So we've learned today that different dietary groups can be identified based on jaw shape. Herbivores have short to medium jaws. Hypercarnivores tend to have short jaws, and mesocarnivores medium jaws. Finally, insectivores tend to have longer jaws. We can also identify different dietary groups based on the mechanical advantage of their jaw closing muscles. Herbivores tend to have high mechanical advantage of both the masseter and the temporalis. Carnivores have low masseter mechanical advantage, but high temporalis mechanical advantage. And insectivores have low mechanical advantage of both muscles. We can also identify dietary groups based on their stress patterns. Herbivores tend to have large areas of low to very low stress and some areas of medium stress. Hypercarnivores have large areas of very low stress, and mesocarnivores have large areas of low to medium stress, of low and medium stress. Finally, insectivores have large areas of medium to high stress. And the great thing is that we have proven that the relationship between jaw shape, jaw function, and diet is the same in today's small mammals and in mammals that lived millions of years ago. So we can use what we know about mammals today to infer diet in fossil mammals. Based on what we've learned, we corroborate that most Mesozoic mammals were probably insectivores, but that there were definitely some Mesozoic mammalian carnivores. And don't forget that my study did not encompass all Mesozoic mammals. We actually left out two big orders of mammals, the multituberculids, and another group I hadn't introduced you to called the haramaids, both of which had a large diversity of dietary preferences. So, while it's true that many Mesozoic mammals were the prey of dinosaurs, not all Mesozoic mammals were cowering under the shadow of dinosaurs. And beyond all the fancy analysis, measurements, and numbers that I just presented, the main thing I would like you to take home is that Mesozoic mammals were very diverse. They lived alongside dinosaurs for over 150 million years, and they were not just hiding underground. They were gliding, they were swimming, they were eating dinosaurs. There really is a world of further work that can be done to truly understand the diversity of Mesozoic mammals. So with that, I would like to thank all of you for listening to me today. And I would like to thank all the wonderful people that helped me carry out my PhD research, including my supervisors, fellow PhD and MSc students, everyone who provided me with data and reviewed my work. Thank you again. And you can find me online on my website, sciencegraphicdesign.com, and all of these various social media places. Thank you.
Well, thank you, Melissa. It was a fantastic talk. Really enjoyed the paleo art and all the illustrations. It was amazing. I just want to send a reminder to everyone that is seeing the talk that you can send questions to our questions eoPERCS host that is Kate today. And I can see that we already have some questions. So I'm going to be copying and pasting these questions to the chat in case you need to read them. So great talk. In the discriminant analysis plot, it looked as if Mesozoic insectivores occupied a much larger morphospace than modern ones. Is there a correct interpretation of that plot? And if so, do you have any thoughts as to why there might be morphological diversity in those earlier, more morphological diversity in those earlier insectivores?
I think that's something that is influencing the position of the Mesozoic mammals a lot in that plot is the phylogeny. So if you were to go into that plot and look from modern mammals are the very earliest mammals. You can even see that the one that we have at the most basal point, which is Haramiyavia, however you pronounce that, actually comes out as a carnivore, which I don't really think is true. But as I said, there is room for error, but it plots right at the top. So I think that what we're seeing there, a lot of it is just phylogeny.
Right, makes sense. Another question is, how did you decide where to apply the loading to your FEA jaw models?
Actually, I didn't just do the first lower molar. I loaded some models at the M1, some models at the jaw tip, some models at the canine. Right now, I chose to present the ones loaded at the first lower molar. They showed the largest difference between different dietary groups. And as for the configuration of the different muscles, I followed the paper by my supervisors where they did FEA on Morganucodon and Cornutherium, and basically used the exact same loading conditions as they did.
Right. Another question is, are there any other analysis of mammal jaws that you want to try?
Yes, actually, another study another set of analysis that I'm going to be starting soon is analyzing the area of the jaws of Mesozoic mammals. So that is indicative of the resistance to torsion and bending in mammals. And in small mammals, it's often been correlated with dietary specialization. We probably don't have as many mammals for this analysis as for the other two, but I still hope that we can get some interesting results out of that.
Right, I don't see any other question coming through, but I do have a question I was thinking about, is that when you answered the first question and you said that there's room for error there when you interpret the discriminant analysis plot. Do you think that for taxa that have teeth available, do you think relying on teeth morphology would be better than relying on jaw shape, for example?
I think that you have to rely on multiple lines of evidence to come to difference. For example, for the large part, what the dietary inferences that I got from jaw shape and mechanical advantage are largely similar to what I got from finite element analysis, for example. But there's definitely been a few discrepancies there. So I wouldn't just rely on one analysis, but look at shape, function, tooth morphology, body size, etc.
Just a final question, I guess, from Jesse. Really enjoyed your talk. I was wondering whether size impacted shape as well. And if so, did you correct for size?
Everything in my analysis is to the same size, so that any differences that I see were just due to shape or to function. But yeah, I mean, yeah, size definitely has a big impact on this, but in the case of my analysis, everything is the exact same size.
Yeah, I think those were all the questions we have so far. So thanks a lot, Melissa. It was a great talk.
Yeah, of course. Thank you.
We're taking the screen share. Just gonna wait a few more seconds if there's any other question. I don't see anything else coming. No, yeah, I don't think so. Thanks anyway. So I'm gonna be taking the screen share.
Yeah, of course.
Right. You can see my screen now, right?
Yeah.
Right. So thank you everyone for coming. Thank you, Melissa, for the talk. Just want to remind people that next week, at the same time, next Tuesday, we have a talk from Advait Jukar from Yale Smithsonian Museum. And he's going to be talking about late Quaternary faunal change in the Indian subcontinent. So join us if you can. And also a reminder that if you know someone that an early career researcher in paleontology that you'd like to nominate, please go to our website and find the nomination form. So now we're going to be transitioning into tea time. That is a good chance for you to learn more about Melissa, her research path, and her work as well. And it's a good chance for meeting new people or talking to old friends, and also for discussing and talking with paleo fans with different backgrounds, and a chance for simply having fun before going back to your regular day. So before we go to tea time, we have a short 2-minute timer, 2-minute break, so you can go to the bathroom, grab a drink of water, and then we can come back and have the tea time. So see you soon.